Project Manager at the International Wheat Genome Sequencing Consortium, and it is my pleasure to serve as moderator for today's monthly webinar series. So welcome everyone. And today we're joined by Andy Chen, who will be speaking about wheat spike and grain development. Before we continue on to Andy's presentation, I'd like to give a quick overview of the IWGSC. As most of you probably know, we're an international nonprofit consortium with more than 3,400 members belonging to over 900 institutes and companies engaged with us from 72 countries. None of this would be possible, our monthly webinar series, other annual workshops, or coordination activities related to genome resources without funding from our 8 sponsors. So I just want to take a moment and thank these sponsors: Arbor Biosciences, BASF, CIMMYT, Florimond Desprez, INRAE, Kansas Wheat, RAGT, and Syngenta. So thank you to our sponsors. As most of you know, the IWGSC was instrumental in the production and release of the first bread wheat reference sequence in 2018. Our mission since that time is to develop additional tools and resources to enhance breeding through an increased understanding of the molecular basis of traits and their allelic diversity. This has included updated versions of the reference genome, updates and refinement of the gene annotation through manual curation effort and functional curation, as well as contribution to the study of landrace diversity. I want to take this opportunity to announce IWGSC events at next month's up and coming PAG meeting in San Diego. As you see here, we'll be hosting 2 workshops, one on Saturday, the main workshop, and then on Tuesday, from structural to functional wheat genomics workshop. And then also on Saturday will be our annual IWGSC business meeting. And so we hope to see you all out there if you're able to attend PAG in San Diego this coming January. Also, please stay tuned for up and coming webinar announcements. If you want to find out about these, you can sign up for our mailing list, which is also shown here on this slide. So I'm just going to give a quick overview of your dashboard. You can ask questions throughout the presentation, but we will respond to the questions at the end of the talk. Depending on how many questions we get, your question may not be asked live, but Andy has kindly agreed to respond to all questions in writing, and we will have a link to those as well as the recorded video posted both at the IWGSC YouTube channel and at our website, wheatgenome.org. If you experience a technical issue, please use the chat. Also, please monitor the chat to see messages from the organizers. Already, you can download this introduction and Andy's presentation as a handout in PDF format if you just go to your control panel. And with that, please welcome Andy Chen. Andy is a PhD candidate in the lab of Dr. Cristobal Wawi at the John Innes Center in the UK. And today he'll be talking to us about insight into the wheat spike and grain development by learning from natural variation and chemically induced mutants. Okay, thank you everybody, and thank you Andy for your participation. And with that, I'll turn it over to Andy.
Yep, thank you, Josh. I'll just get my screen set up. Let's see. Josh, you can see my screen okay? And I'll get started.
It looks good.
All righty. So yeah, thank you, Josh, again. And then thank you, the organizer, for an opportunity. Sorry, just setting up my screen a little bit. For the opportunities to speak at this webinar. So today I'll kind of showcase how we have applied the resources that were developed by IWGSC and the community to help us better understand genes or allelic diversity that can control spike and grain development in wheat. All right. So as the population continues to rise, there's an increasing need to stabilize or even increase food production. So here's a picture of wheat and its related subspecies. So you can see very striking difference with their color, their size, and their morphology. So the main goal of my project is to understand what makes them different from one another, and can we use this source of diversity to better understand wheat development and identify strategy to improve yield components, right? So the wheat spike is an inflorescence with very short branches, and it is where all the grains are produced. So traits such as the number of grains produced per spike and the size and weight of the grains are important components of the final grain yield. And so the first part of my talk, I'll be focusing on spike development. So between terminal spikelet stage, well, this is where all the spikelets are formed, and anthesis, there's a significant change to the length of the wheat spike due to the elongation of the rachis. So the spike length kind of reach max length around yellow anther stage, and most of the spike and rachis elongation happen before that stage. So you can see a very dramatic changes in spike length. So in terms of spike length, there are many loci that have been identified to play a key role. So for example, the dominant C locus is responsible for the morphology, the compact spike morphology of the club wheats. As you can see, there's about 50% decrease in spike length. And this is a very characteristic of the wheat subspecies of compactum. This locus has recently been fine mapped to an 11 million base pair region from 231 to 242 million base pair on chromosome 2D. But the investigation for the underlying gene I think is still ongoing. And another locus is the Q locus, which is involved or very important for the domestication of wheat and is linked to the free threshing characteristic of the modern wheat. But in addition to that, it also has some effects on spike length as well. So the ancestral lowercase qq confers a speltoid phenotype. So you can see the spike is very lax and longer, whereas the dominant Q allele, which is common across modern wheats, gives a semi-compact phenotype, as you can see in this photo. So Q is AP2 gene, and the based on nomenclature is called AP2L-A5. And so AP2L genes are regulated post-transcriptionally by microRNA 172, so or MIR 172 here, which would target the AP2L mRNA for cleavage and degradation. And this interaction is very dependent on the sequence being complementary to one another. So here I'm showcasing the uppercase Q allele carries a mutation in the microRNA 172 target site from C to T, and this then introduces a mismatch with the microRNA, right? And then which will then increase the AP208 expression, leading to the subcompact phenotype that we are looking at. So our idea then at the beginning was then to identify genes other than the well-known locus I've talked about that can influence spike length to better understand the genetic basis that control this trait and the allelic diversity that we can apply to modify this trait in the field. So for our collaborators, we're able to get 3 sets of near-isogenic of the hexaploid wheat N67. So they're all genetically very similar, except for that locus that gives them the compact spike phenotype. And these lines, I just need to mention that these lines are developed a very long time ago and are developed simply by backcrossing to N67 and selecting for that compact spike phenotype. So you can see here, NK15 is slightly shorter. And another characteristic of NK15 is that the distance between the top spikelets are a lot shorter as well. And the spike length phenotype is a lot more drastic on line 2 and 3, both very, very short, more than 50%, around 40 to 50% decrease in spike length. So we first characterized the stem elongation process between the near-isogenic line. And what we have found is that at the white anther stage, which is very early on during the stem elongation phase that I've just mentioned, all of them are very similar in length. But as early as the green anther state, we see that line 2 and 3 are already shorter, while ANK15 is still very similar to N67. And we don't see the difference between N67 and ANK15 in terms of the spike length until the heading stage. And one of the advantages of working with near-isogenic line is that they're very similar in genetic background. So our approach was initially to use RNA sequencing to kind of narrow down some of the candidate genes that we want to look at. And as you see from this photo, most of the elongation, at least for ANK15 and N67, occur between the green anther and heading. So our hypothesis was that the causal gene for the shorter spike phenotype must be differentially expressed at this stage. So we then collect rachis from 4 independent plants and send them off for RNA sequencing. And today my presentation will focus on the work that we have done for ANK15. All right, so a little background on ANK15. The compact spike locus was introduced from a mutant line of wheat cultivar scale-up, and the compact spike phenotype has been proposed to be controlled by a single locus on chromosome 2b based on this study in 2016. So since they're near-isogenic, like we expect them to be genetically very similar, except for the locus that controls spike length. So the very first thing we do is we were looking for single nucleotide polymorphism difference between NCC7 and ANK15, and we did so by aligning the RNA sequencing data against RefSeq version 1.0 and compare them against one another. So each one of these color line in green is representing one SNP. So you can see that most of the genomic region are not different, right? They're white, so they're not different from one another, supporting that they are near-isogenic in nature. But what was very surprising to us was that the putative region in the study from 2016, which is highlighted in purple, there was no detectable SNPs in this region between the two lines. And the closest one was about, I think, 100 million base pair away. And in fact, what you can probably notice is that majority of the polymorphism is on chromosome 5A. And there's some polymorphism on 5D, 7B as well. So we're a little bit worried at this point. But we still follow up with the differential gene expression analysis. So we perform differential gene expression analysis and look for genes that are differentially expressed. And one of the genes that caught our eyes when we were doing this analysis was AP2LD5, which is the D homolog of the Q gene that I've just mentioned earlier. So we found that based on the RNA-seq data set, we found that NK15 carries a single nucleotide polymorphism at the MIR172 binding sites right here. And this SNPs introduces a mismatch, and based on what we know about the mechanism, this can potentially influence the cleavage efficiency and increase the expression of AP2LD5. And we saw that in our RNA-seq data set, right? So ANK15 had higher AP2LD5 expression. So we repeat that experiment. We confirmed that by performing qRT-PCR and show that AP2LD5 indeed had higher expression in ANK15. So previous study had shown that AP2LD5 plays a role in the suppression of the speltoid phenotype. So in this study, they knocked out this gene and they found that you get these speltoid-like spikes, so a very lax spike. So we know that it plays a role in that in spike development. And in fact, a study in 2018 has proposed that it is the underlying gene for RHT23, which is a locus that confers reduction in height and also compact spike as well. So you can see that the mutation of RHD23 was just a few base pair away from ANK15. So they appear to be working in a very similar mechanism where there's a SNP in the microRNA binding site. So it is also worth noting that ANK15 was a bit shorter in the field as well. So it also has a reduced height phenotype. But it's quite interesting that RHD23 with a different mutation was able to have a more drastic spike length phenotype. But given what we know of these evidence, what we know so far, AP2LD5 was our primary candidate gene at this part of our study. So to test that hypothesis, we then generate an F2 mapping population between NK15 and N67. So we perform QTL mapping. So first we have markers on chromosome 2B because they're near-isogenic in nature, so there were limited amount of SNPs in which we can design markers on. So this purple region is where the previous study has proposed the locus to be, but we saw no association between the 3 markers that we designed and the spike length phenotype. And essentially, this graph will show you the strength of the association between marker and the phenotype. So we then designed additional markers for chromosome 5A, and this was the region that was highly polymorphic between NCC7 and ANK15. Again, we're not seeing any association between the markers and the phenotype, and this is where Q is located. So this would suggest that the locus is not on chromosome 5A. And lastly, we designed markers on chromosome 5D around the AP2LD5 region. And in fact, the highlighted marker was designed based on the SNP in the microRNA 172 binding site. So we see a very strong association, we see a very strong and significant association between these markers and the spike length. And the F2 lines with the N67 allele has longer spike in comparison to the F2 lines with the ANK15 allele. So given our mapping data, our differential gene expression analysis, and what we know about the function of AP2LD5, we propose that AP2LD5 is likely the gene underlying the compact spike phenotype of ANK15. So that as I was mentioning before, we were hoping to identify novel genes that can affect spike development. So we were quite surprised by the results of ANK15. But that's just part of research, and we were still able to apply the tools that IWGSC has developed and the community has developed, and to elucidate it and identify a new allele that can control the spike length morphology. So yeah, so the first part of the study, I talk a little bit about wheat spike. So I'm going to shift gear and talk a little more about grain development and how we have used subspecies of wheat to better understand grain development in wheat. So each spike contains these 4 bearing branches known as the spikelets, and each spikelet is subtended by 2 outer bracts known as the glume, and also contain multiple florets, which are floral structures that can bear that have the potential to bear 1 grain each. And within each floret, there are 2 opposing maternal floral structures known as the lemma and polea. And these 2 structures will kind of envelop the grains, envelop the carpel as it develops into grains. So since lemma and polea envelop the grain during development, many studies have hypothesized that the space within these 2 structures can limit grain growth and ultimately determine the final grain size. So in both rice and wheat, and especially in rice, we have seen many genes that control the size of these 4 organs being the underlying gene of major QTL for grain size. So this would suggest that by altering the size of these structural organs, we can also modify grain size as well. And this is what motivates us to look at natural variation of the size of these organs within wheat subspecies. So these are some of the previously identified loci that affect glume size and floral organ size. So for example, this is one of my favorite examples. The tunica locus in pot corn led to an elongation of glume in corn. So here on the top, you see wild-type corn, where you can see the kernel, but in the popcorn, the glues are so elongated to an extent that it completely covers up the kernel. And the underlying gene was identified to be ZMM19. And the P1 locus in wheat from Triticum polonicum also led to an increase in not only the glume, but lima and grain as well. The underlying gene was identified to be VRT-A2 by the work that was done by Nikolai M. James from a group, and also independently at China Agricultural University in Beijing. And Nikolai was able to give a talk on P1 about a year ago on this webinar as well. So if you haven't had the chance already, I would recommend checking out YouTube channel. I think he gave a really good talk on this topic. And the last one is the P2 locus from Triticum ispahanicum, right? It also has elongated glume, lima, polea, and grain as well. So my presentation today will focus on some of the work that we have done so far with Triticum ispahanicum. So Triticum ispahanicum is a tetraploid wheat that was first identified in the Ispahan province of Iran, hence the name Triticum ispahanicum. And the phenotypic difference between Triticum ispahanicum and other tetraploid is quite drastic. So here we are using Langdon, another tetraploid wheat, as an example. And then you can see that Triticum ispahanicum has these longer glume phenotype and also appear to have longer spike as well. And then this longer glume tray is controlled by one major QTL known as P2. So we first want to characterize how this P2 locus affects yield-related trait by using near-isogenic line that was generated by a collaborator in Japan. So the wild type is depicted in blue, while the near-isogenic line carrying the P2 is in orange. So in the LD222 and semi-dwarf LD222 background, we observed that similar to the full organ, introgression of P2 leads to an increase in grain length. In addition, P2 is associated with an increase in spike length, which is accompanied by an increase in spiker number when compared against the wild type that is in blue. However, this increase in spiker number did not translate to an increase in the number of grains per spike as we would hope. In fact, we have observed a decrease in the number of grains per spike in the near-isogenic line with the P2 allele. And this is because P2 decreases the number of fertile florets, or the number of grains produced per spikelet. But nevertheless, we're still quite interested in the underlying mechanism of P2 that lets it modify grain size. So the work that was started by our collaborator has previously located Triticum ispahanicum, the P2 locus, onto chromosome 7b by investigating the phenotypic linkage of the long glume phenotype with other known traits on chromosome 7b. So very classic linkage experiments. And some study have speculated that the P2 is homologous to the Triticum polonicum P1 locus on chromosome 7a, right? So they're both on chromosome group 7, and they share very similar phenotype in terms of their elongated glume, their elongated lima. So as I was mentioning, Nick from our group has found that VRT-A2 was the causal gene of P1. So we therefore hypothesized, we asked the question whether or not the causal gene of P2 is in fact the B copy VRT-B2, given how similar their phenotypes were. So to test that hypothesis, we have developed a F2 mapping population between Langdon and Triticum ispahanicum sessions. And these two lines were shown in the previous slides where they're very different in terms of the size, the length of their group. So the wheat reference genome has enabled us to rapidly develop genetic markers. We identify Triticum ispahanicum specific polymorphism by mapping some of the previously published RNA-seq data set against the Chinese Spring reference genome. And we have designed 13 markers spanning across chromosome 7b. So we perform QT analysis and look at whether any of these markers have an association with the length of the glume. And again, this is the diagram just shows the strength of the association. And to our surprise, none of the markers on chromosome 7b has an association with glume size. And here is where our original candidate is located, VRT-B2 is located. So this result contradict with previous hypothesis and show that P2 is not on chromosome 7b and VRTB2 is likely not the gene that we're looking for. And we therefore develop additional Cas markers on all the other chromosomes. And we find a significant association between markers on chromosome 6a and glume length. And the marker with the strongest association is on 549 million base pair. And you can, in this diagram, you can see that the F2 lines that carry the Langdon allele at the marker has shorter glume than the F2 lines with the ispahanicum allele, and the heterozygous is somewhere in between. So these results show that, contrary to previous study, the P2 locus is on chromosome 6a instead of 7b. To follow up on this initial mapping, we use homozygous recombinant to further fine map the P2 region to an interval of 1.68 million base pair. And based on the IWGSC RefSeq version 1.1 gene model annotation, there are 10 high confidence genes within this interval. This includes GTP binding protein, I think ubiquitin protein transferase, and more. But we have identified SVP-A1 at that stage as a strong candidate, and I'll explain why that is. So short vegetative phase, or SVP genes, belong to a subfamily of Matchbox transcription factor and have important function in controlling flowering time and also floral organ identity as well. And we see that in Arabidopsis and also in some of the recent work in weeds. And this is a phylogenetic tree of SVP protein in rice, wheat, and maize. So in grasses, there's a triplication of SVP gene, resulting in 3 distinct clades as highlighted by these boxes. And natural variation in genes from SVP family have resulted in drastic changes in the size of their glume tissue. So for example, like I was saying, the causal gene for the long glume phenotype in Triticum polonicum is VRTA2, and VRTA2 is paralog to SVP-A1. And the causal gene for the elongated glume in popcorn ZMM19 is a direct homolog of SVP1 in maize. So given this previous connection between SVP family gene and elongated glume tissue, we think SVP-A1 is a strong candidate within our fine map region. So we therefore go on to characterize SVP-A1 allelic variation in Triticum ispahanicum. So to do so, we use a whole genome sequencing data of Triticum ispahanicum. So I want to give special thanks to Fei Lu's group from IGDB of Chinese Academy of Science for giving us early access to this data set, and they have since published the story in 2020. So we aligned the sequencing data against the reference Chinese spring genome to identify polymorphism within SVP-A1 gene. So we have found a 482 base pair promoter deletion, 53 polymorphism in the non-coding region, and 1 missense mutation on exon 5. So we're particularly interested in the promoter deletion, as we hypothesize deletion of this size can likely influence the expression pattern and expression level of the gene. So we later Sanger sequenced the promoter region of SVP-A1 of 7 independent ispahanicum accessions and confirmed that this deletion is present in all of them. And we also developed a Cas marker for the promoter deletion. And Julian Zhang from UC Davis has screened over 300 different tetraploid wheat and have found that the promoter deletion was not present in those other tetraploid wheat. This would suggest that the promoter deletion is unique to Triticum ispahanicum, making it a strong candidate as a causal polymorphism. So we first look at the expression pattern of the wild-type SVP-A1 across different tissue using the wheat EFP browser. This is based on the RNA sequencing data of wheat development of the hexaploid wheat accession Azanaya. And the tissue color in yellow has no expression, while the tissue color in red has higher expression. So it seems like SVP-A1 is expressed in most of the tissue, right? It's expressed in developing meristem, it's expressed in flag leaf, it's also expressed very highly in the anther tissues, but however, SVP-A1 is not expressed in lima and glume. Remember, these are the 2 tissues that are elongated in Triticum ispahanicum. So we therefore want to investigate how does this promoter deletion in SVP-A1 influence the SVP-A1 expression pattern. So we again go back to that near-isogenic line that we have talked about, where the wild type do not carry the promoter deletion, while the line with P2 introgression has that promoter deletion. So first we look at SVPA1 through meristem development at double ridge, glume primordium, and terminal spicular stage. So in wild type, SVPA1 is expressed in these tissues during all the stages that we have looked at. And in the ispahanicum line, with P2 introgression, we observe a higher expression of SVPA1 at these stages, with difference in glume primordium being borderline statistically significant. So we have also looked at other tissues during the late brooding stage. Right, so we found that in wild type wild type, which is in blue again, we found that SVPA1 is expressed in flag leaf, is expressed in anther, whereas no detectable or low expression in glume, lemma, and pollia. And so this agrees with the RNA-seq data set that we have just looked at. However, in the near-isogenic line with the ispahanicum allele SVPA1, we see significantly higher orthotopic expression of SVPA1 in glume, lemma, and pollia. Remember, these are the tissues that are elongated in Triticum ispahanicum. So in short, the P2 from ispahanicum and the 482 base pair promoter deletion is associated with an increase in SVPA1 expression across multiple tissues. So since the promoter deletion is associated with a change in expression, we hypothesize that within this 482 base pair, there must be some regulatory motif within the deleted region. And sequence with regulatory functions are more likely to be conserved across evolution. We therefore align the promoter sequence of SVPA1 with its ortholog in other grass species, including I think Brachypodium, barley, rice, sorghum, and maize. And we look for evidence of conservation with a program called MVista. So we set our conservation threshold to look at 20 base pair and 85% sequence identity. And the red region are windows that have passed our threshold and our region that show some conservation signature. So we found two motifs, M1 and M2. They are not only conserved in sequence, but are also positionally conserved across grasses, meaning they are the same region of the promoter. And we also use the MEME suites to search for sequence that are not positionally conserved, but only conserved in sequence. So in addition to M1 and M2, the MEME suite was able to pick up M3 that's conserved in sequence, but not locating at the same part of promoter across grasses. So conservation of these sequences would suggest that these motifs might have some regulatory function. So our hypothesis at this point is that promoter deletion of these motifs then is what led to the misregulation of SVPA1 expression. So to test this hypothesis, we use CRISPR-Cas9 to edit the promoter region of SVPA1. So we have designed 3 separate guide RNA in order to generate different promoter allele, either with whole region deleted, like we observed on Triticum ispahanicum. We named the allele SVPA1b, or just some partial promoter deletion, which can help us dissect out the function of these conserved motifs. And in our construct, we also use a chimeric protein GRF in order to improve regeneration efficiency of the plants. So we were able to recover different alleles in our T2, but today I'll focus on event F.2 and F.8. So event F.2 has a ISP-like deletion as the 472 base pair removing all the conserved motif, whereas event F.8 have partial deletion, just removing motif 2 and 3. So we want to look at how these promoter deletion affects the expression pattern. So event F.2 and F.8 in wild type sister lines that do not carry the promoter deletion, we see no to low expression of SVPA1, very similar to what we have observed in the near-isogenic line. However, in event F.2 that has the 3 motif deleted, we see increase in SVPA1 expression, very similar to what we have observed in the near-isogenic line. And for event F.8, we also see a change in expression level, albeit at a lower level. So the next thing we look at is whether this increase in expression via promoter deletion is then sufficient to modify glume length. So we look at the glume across the entire spike. In this case, we focus on event F.2, and we found that across the entire spike, you can see event F.2 has a longer glume, similar to what we have found in Triticum and ispahanicum, and this shows that the promoter deletion is sufficient to increase glume length. So we have also looked at other tissue. Again, lemma and polea are the two tissues that are encapsulating the grain as it develops, and we can see that they're also longer. And more excitingly, we also see an increase in grain length across the entire spike as well. So this is so we're quite excited about it. So these work were all done in hexaploid wheat in fielder. So we want to see whether or not we can see a similar thing in tetraploid wheat, which we use Chronos in this case. So we use a very similar approach, 3 guides, and was able to generate 2 events, event K.2 and event K.4, which are 2 partial promoter deletion. And again, we're seeing this increase in SVP-A1 expression in the glume tissue of these events with partial promoter deletion. And these partial promoter deletion was sufficient to increase glume length in a tetraploid background, right? So it looks longer, it's also a little wider as well. But one of the stuff that I didn't bring up earlier was that this partial promoter deletion was not sufficient to increase glume length in hexaploid. And we can talk a little bit more about that later on. But essentially what these results is showing us is that SVP-A1 is the underlying gene for the long glume locus P2 from Triticum ispahanicum. And also these identified motifs are required for the regulation of SVP-A1 expression in the glume tissue. Right. So this is a slide that I've showed earlier on. We show that these motifs are conserved in other grasses, right? So it's also conserved in barley, it's conserved in rice. So our next question was, since we know that these motif functions to regulate SVP-A1 expression in wheats, do they have similar function in other grasses such as barley as well? So we look. So our next part of the study will focus on barley because in terms of spike characteristic, it's very similar. It's more similar to wheat as opposed to maize. Right. So we also use a similar approach. We use CRISPR-Cas9 to target the promoter region of BM10, which is the closest ortholog to SVP-A1. And in BM10 of Golden Promise, which is the cultivar that we're using, they also have the 3 conserved motifs that we have identified. And with this, we're able to identify 2 independent events, GP1 and GP2. So GP1, we have very similar to the partial promoter deletion events, with motif 2 and 3 deleted. Also remove parts of 5' UTR. And we also have GP2, which remove some sequence upstream of motif 1. Okay, so when we're looking at T-Knot plants, we saw GP1 and we were quite excited about it because in comparison to the Golden Promise that was grown as control next to it, it is a lot longer, and it looks like the seeds are longer. It looks like the lemma tissues are longer in general. So we initially were quite excited about it, but unfortunately, when we try to follow it up in the T1 generation, what we have observed is that there were no change in the BM10 expression comparing the wild type, which did not have the promoter deletion, in comparison against the mutant. Right? So they have very similar expression pattern of BM10, and BM1 and VRT2 are paralogs of the BM10 in barley. So this promoter deletion did not lead to a change in expression of BM10 in the lemma tissue. And we have also looked at grain length and also spike length of these events, of GP1 events. And for now, we did not see a difference in grain length between the mutants and the wild type. And we also did not see a difference in spike length. However, so based on our preliminary result, this would suggest that perhaps BM10 is regulated a little differently in SVP-A1 in wheats, and perhaps these motifs do not have conserved function across different species. However, I want to illustrate these was just based on one single event, So we need to identify additional events with different deletions to make sure that these results are actually right, are not just an artifact that we're seeing in this event. Okay. So to kind of sum up the presentation. So we have seen that natural variation at cis-regulatory region of SVP transcription factors being the underlying polymorphism of major QTL, QTL of floral organ and grain size and cereal. So in this study, we found that promoter deletion is the cause of polymorphism for the long glume phenotype in Triticum ispahanicum, right? And the pot cone in maize, again, is known for having the elongated glume and is controlled by the tunicate locus. They have found that this is caused by an ectopic and high expression of CMN19, which is an ortholog of SVP1 in maize. One thing that was interesting was that in pot cone, they also found duplication and promoter rearrangement of CMN19. And this promoter rearrangement, in fact, removed the 3 concern motifs that were within the deleted region of Triticum ispahanicum, suggesting maybe there's some kind of similar under mechanism that are in play. And lastly, the respective parallel VRT2 with the intron-1 rearrangement was found to be the underlying gene for the P1 locus. So these examples demonstrate that the change in expression of the gene, for the P1 locus is due to intron-1 rearrangement. And these examples demonstrate that the change in expression of the gene from SVP subfamily can generate quantitative difference in the size of these 4 organs and even grain length as well. And I found this quite fascinating because SVP was first characterized due to its involvement in flowering time. So I guess looking forward, can we then learn from these examples and engineer variation in cis-regulatory region for developmental genes in polyploid wheat in order to generate quantitative improvement in these EO-related traits? I think we can, right? Because first of all, with the development of the resources of wheat genomic resources and annotated genome, we're getting a better understanding of what these key developmental genes might be. Furthermore, cis-regulatory variation already underlie many of these agronomically important alleles, whether it is to control virilization response or control photoperiod sensitivity. And we're also getting more and more tools that can help us generate these novel variation in cis-regulatory region. So I've illustrated that we've used CRISPR-Cas9, but recently work led by Juning Zheng in UC Davis has used promoter capture to sequence and identify EMS mutants of chronos that also carry polymorphism at these putative cis-regulatory elements. So this is another tool that can complement CRISPR to help us generate variation in these regions that can affect agronomically important traits. And we also have, a lot of public data sets that are coming out these recent years that are characterizing open chromatin region using ATAC-seq and also looking at histone modification. And these public data set essentially can help us kind of pinpoint which cis-regulatory element might have a role in regulating the expression of this gene and help us focus on effort in those more important regions. And last but not least, there has been a change in legislation and change in attitude towards gene-edited plants. Right. For example, in the UK, we have passed the genetic technology bill into law, I think just this year. And what this allows us to do is that we are now able to test the gene-edited transgene plants in the field and really bring our research in the field and see how they would behave, which I think is a very important part of research to hopefully bridge that gap between research and application. So this year we were able to put in Fielder and our event F.2 and F.8 in the field thanks to this change in legislation. You can see our team, Pam, Nick, and James and Nick working really hard to set up these plots. And we're also seeing a change in attitude in Canada as well, right? So these are a statement made by Health Canada and CFIA, which is the Canadian Food Inspection Agency. But essentially, what they're trying to say is that gene-edited plants should be regulated like all other products of plant breeding, right? And they should be more focused on their final characteristic and not the method of product development. So this change in attitude can potentially, a change in attitude and change in legislation can potentially promote the use of these CRISPR-Cas9 technology to generate cis-regulatory alleles that might be beneficial agronomically. So I think the future is looking very, very promising. Okay. And with that, I would like to acknowledge people that were involved made this work possible. So of course, my awesome supervisors, Chris Wawi and Nikolai Adamski, and also our collaborator, Nobuyoshi Watanabe, who initiated the work on P2 about 20 years ago and has generously supplied us with the near-isogenic line he had developed. And these near-isogenic line, they were backcross 6, backcross A, so they take many years to develop in the past. And I also want to highlight the contribution of our previous master student, Victoria Zhang, who has done a tremendous work with ANK15. So if you have time and if you're interested, please do check out her preprints bioarchive. And also I want to highlight the contribution of the IWGSC community for the resources they have developed, right? The study would not have been possible, would have been a lot more difficult without these resources. And hopefully in alignment with the vision of the version 2 alignment, we can apply these resources to identify allelic variation or have a better understanding of genetic basis that are controlling some of these traits that we're interested in. And thank you for the funders for my PhD, of course, the Natural Science and Engineering Research Council of Canada, and also thanks to John Innes Center and John Innes Foundation for funding my research. And yeah, thank you all for listening, and I'll be more than happy to take questions now.
Here's a question from Ruben Vicente. Do you think there would be any compensation mechanism in modern wheat if larger glumes were introduced?
Yeah, that's a possibility. So we in fact have looked at introducing P1 into a modern wheat, a UK modern wheat paragon. And what we have saw so far is that it didn't have any penalty on thousand kernel weights, and it doesn't seem to have any penalty in terms of other EO components in the field. So Ruben, based on what we can see so far, it doesn't seem to be a trade-off in terms of the P1 locus work that was done by Nikolai. However, that being said, when we introduce the P2 locus, which comes into a tetraploid, and we look at these near-isogenic, we're seeing a little bit of penalty in terms of the number of grains produced per spike. And I'm at this point, I'm not 100% sure whether that is due to a elongation of the glume or because we're getting bigger grain as a result of introducing a P2 locus. But yeah, you're right on the spot. We do see some compensatory effects on other EO component traits. Yep.
So maybe you get larger grains but fewer in
That's right. Yep.
Okay. All right. The next question is from Yelena Beneventi. Nice talk. Just a basic question. Chromosomal rearrangements compared to Chinese spring have been described in many wheat cultivars and may also exist in tetraploid wheats. Could this be somehow responsible for differences in loci mapping between yours and previous reports? Yeah, so it seemed like you had at least one, maybe two cases where a trait had been mapped in the literature, and then you go and look for it, and it's not there. Yeah.
Yeah. Thank you, Lena, for the question. I at this point, I don't think that is the case. I think this also highlights some of the resources that IWGSC has developed, right? So now we have annotated genome, which allows us to design markers that can more accurately pinpoint where these traits might be. And because these studies were conducted 20 or 30 years ago, so I think it might just have been a mistake on their parts that it had happened. We're not sure why it had happened, but based on the near-isogenic line that we have looked at, it seems like they were proper near-isogenic line. It's just that the genetic work, maybe without the markers that we have right now, will end up giving results that is not consistent with the finding that we have. Yeah.
So you have much better markers, more specific markers.
Yeah, I think so. I think that's one of the contributing factors. Yeah.
Okay, thank you. Okay, this is a question from hopefully I pronounced this right, Bihendra Kadka. Congratulations on your excellent talk. Curious to know if you have any hypothesis for the mode of function for SNP found in the microRNA MIR172.
Yeah, thank you for the question. So in this case, we think that the mode of action is that this SNP introduces mismatch with the microRNA, and then therefore, since this interaction is very dependent on the sequence being complementary to one another, we think this then reduces the cleavage efficiency, and therefore we see cleavage efficiency of the mRNA, and therefore we see a higher expression of AP2LD5, and we think this is what leads to the shorter spike phenotype that we see in AMK15.
Okay, and then this is the final question from Ahmed Charif. Have you looked up if the QTL or gene you mapped here is associated with some other traits like disease resistance?
Oh, okay. So thank you, Ahmed, for the question. That's a good point, right? Because when you have longer glume, you're generating these type of microenvironment that might be more humid, and that might then lead to more susceptibility to certain disease that requires a humidity to happen. No, we have not looked at them in depth, but that's something that's something that would be quite interesting to ask. Yeah. So I don't know the answer to that question at the moment.
So I wonder if probably the asker knows the answer to this question, but has there generally been cases seen where a more compact type of spike can lead to, say, greater fungal diseases or things like that?
Yeah, no, that's a good question. I haven't had the chance to explore that area yet. I don't know the answer to that. Yeah.
Okay. Well, that was the end of our questions. So I think we can wrap it up. We have less than 2 minutes left. So Andy, are there any final thoughts you'd like to give to wrap up with?
No, but I just want to thank everyone for coming to the seminar. And then again, I just want to highlight the contribution from the resources that were generated as a result of IWGSC, and also all the people. Just want to thank all the people that have contributed to this work. And also on the personal notes, I've recently started a new job as a wheat breeder in Lima Grain in Canada. So looking forward to apply some of these work that we have done into my own breeding pipeline in the future as well.
Well, congratulations. That's terrific. We're glad to hear about that. And I certainly want to second everything that Andy said in terms of your participation. Thank you so much. We had quite a few, over 110 participants today. Just to note, for people who might have missed this or want to watch it again, the entire webinar has been recorded and will be posted soon on our YouTube channel. So look out for that, and look out for future webinar announcements. And thank you again so much, Andy Chen. Yeah, so with that, goodbye, everybody. Enjoy your day. Thank you.